So as we talked about these biomolecules throughout the course, we learned that each of the biomolecules were made of these subunits. And these subunits put together in chains made up these things called polymers. So these polymers were made of monomers. So finally, the subunits are called monomers, and they make polymers. Now, the polymer is just this idea of this long chain molecule made of subunits, and these subunits, again, depending on which the subunit is, defines this larger molecule. So if you had a bunch of amino acids put together, you'd call it a protein. If you had a bunch of nucleic acids put together, you'd call it a DNA or RNA molecule. So here we're going to look at and focus in on these nucleic acids. And these nucleic acids there's two types that we talked about. And we talked about some other uses and some other concepts that kind of define the nucleic acid. And we looked at the portions of the nucleic acid that were acid. And we broke up the nucleic acid into its monomers. And the monomers, we said, were nucleotides. And each of the nucleotides were composed of three parts: one, the phosphate; two, the ribose; and three, the nitrogenous base. The same thing goes for the RNA molecule, which is made of, again, this phosphate, this ribose sugar, and this nitrogenous base. And the nitrogenous bases had four different kinds. You had the A, the T, the C, and the G. So depending on which nitrogenous base, either pyrimidine or purine, was attached here to the ribose sugar, that defined what this nucleotide was. And so you could look at a chain of these and say, okay, this DNA is A, T, C, G, C, C, T, C. So when we looked at the sequence, the DNA sequence, what we're looking at is what is the sequence of monomers that are attached to one another that define this nucleic acid. The same thing goes then with the RNA, except that with RNA, instead of having a T, you have a U. Now all this is described in the audio, I believe. Unfortunately, we lost half the audio in a technical glitch, and we lost some photos, so I'm kind of touching on it. I need you to remember that all this is in the notes. We covered it already, and don't panic. We're hitting only the key points, which is what are the parts of the nitrogenous bases, the fact that there are two types of nitrogenous bases, DNA and RNA, and the fact that you have four different possibilities for the nitrogenous base portion of the nucleotide, and that, again, those nucleotides could either be DNA or RNA, the difference being that DNA is missing an oxygen where RNA has the oxygen. Kind of pretty straightforward. When you're looking at nucleic acids, DNA has pretty much two, and depending if you're looking at if you're talking about the nucleotide or the polymer, there's two or three different functions. First is genetic information, so it carries this stuff that's the stuff of heredity. So everything that makes up who you are, that's inheritable, is due to DNA. It's the informational packet. So this really does carry information. And remember, we talked about what information was, which is differences. So anytime you have a series of differences, then you have information. When we look at what nucleic acids can be used for, remember that these nucleotides can form, instead of having one phosphate, if you have three of them connected to it, then you could have something called ATP or GTP, and that could be used during respiration or photosynthesis. Now again, respiration, photosynthesis, we covered it in class. It's testable material. It's in the notes. But I'm not going to ask you big ideas. I mean, I'm going to ask you the big ideas, not the details. Okay, so I'm not going to ask you the enzymes involved in the respiration process. What I want to know is, do you understand what the three phases were? But we'll talk about that when I send you the respiration video. So nucleic acids, DNA, it's an informational molecule. It's a long polymer. It's made of these nucleotides, these monomers called nucleotides. There are four of them, A, T, C, G, and can be used as energy. If it's a nucleotide with three phosphates on it, it can be used as energy, and it is used as energy as the main currency, energy currency of the cell. Of course, the method by which this genetic information can be translated into functional units is that this DNA molecule, and different portions of it are what we call genes, and these genes code for protein. All right, so that's DNA in a nutshell. RNA is even a little bit more versatile. It carries information in the form of a messenger. It takes the DNA message from DNA over to to the ribosome. But RNA also makes up the ribosome. So I sent you a video on RNA, on ribosomes. I believe it was June Kane that asked for an explanation of ribosomes. So I found a nice video that actually saw the process of translation. So you saw the mRNA leaving the nucleus and entering the cytoplasm where the ribosomes attached and made the proteins. So when you're looking at RNA, it has an informational purpose, has an informational function, but it also has an actual functional. It actually functions as an enzyme in the cell. So it's not just informational, but it's also functional when we're looking at the purpose for this molecule. Again, I just want to touch on again that it's made of nucleotides, that it has phosphate, it has nitrogenous base, it has a ribose sugar, that the difference is that this ribose, or somebody asked me in class, a pentose sugar, that this pentose sugar has an OH group, a hydroxide group, and because of that we call it RNA. When it doesn't have it, we call it DNA. And that's pretty much the concept behind nucleic acids. Of course, we still have translation and transcription and translation to touching on it. And remember the energy usage at ATP, that whole thing of respiration and photosynthesis. All right, so that takes us through this video, and we'll see. You'll hear from me again in just a moment.